Ming Rui is going to give us a talk on functional connectome features and how to use them to study psychiatric disorders. Take it away.
Thank you very much. Can you see my screen?
Yes, I can.
Okay. It's my great pleasure to join this wonderful meeting. And Robert and Fernanda have shared their wonderful models and the softwares. So my talk will be a little different. I will mainly focus on studying the alterations of functional connectome features in psychiatric disorder. Well, as we know, the human brain is a very complex system. It works as the fundamental biological basis to support various cognitive performances. So different brain regions are interconnected by white matter fibers and synapses. The human connectomics provide an important research framework to study the connectivity pattern and organization principles of the human brain. So the human connectome or brain networks can be described at different spatial scales, ranging from the microscopic neural level to the macroscopic regional level. So over the past 3 decades, the multimodal MRI techniques developed very fast, both for structural and functional imaging. It provided invaluable techniques to examine the anatomy and activity patterns of the human brain. Because the MRI imaging are non-invasive, in vivo, and repeatable measurements, they are particularly beneficial for investigating the brains of healthy people and patients with mental illness. More importantly, to date, the accumulations of multi-center MRI big data offer us a great sources to investigate the brain networks. So with different modalities of MRI data, for example, the resting state fMRI data, the structural or diffusion MRI data, we can extract different information from the brain, just like the brain activities, the structural morphologies, or white matter fiber tracks from different brain regions. By estimating the correlations or fiber connections between each pair of the regions, we can therefore construct the functional or structural brain network models at macro scale. Then we can use graph theoretical methods to review the architecture of the brain networks. For example, at the global level, we can examine whether the brain networks follow a small world configuration with a balance between the global efficiency and local efficiency. We can also identify some important hub nodes with dense connections or some pivotal edges for information transfer in the brain network. We can also assess the modular structure of the brain network and recognize some connector hubs that are critical to link different models or different systems. To analyze these brain networks based on MRI data, during the last 10 years, we have developed a series of toolboxes and codes. These include DIRED, which is a deep learning-based framework for multi-center MRI imaging harmonization. Gritner is a graph theoretical analysis toolbox for analyze the brain networks. The CCAT is an easy-using tool to conduct both seed-based functional connectivity analysis and the network connectivity strength analysis. The Pagani is a CPU-GPU hybrid framework for high-resolution brain network analysis. It can deal with a large network having over 1 million nodes in the network. And also, we have the visualization toolbox, BrainNet Viewer, and some other codes. all these toolboxes and codes are freely available for researchers to conduct an imaging studies. So with the help of these tools, we have reviewed some typical features of the functional brain networks in healthy subjects, such as we found the small worldness configuration are reproducible between two scans with a long interval, for example, 6 weeks. We have identified some hub regions with dense connectivity, mainly located in the medial and lateral frontal and the parietal regions. And the white matter fibers can connect these regions, are hub regions, pivotal edges for information transfer in the structural brain networks. The functional network also exhibits a significant modular architecture, both at the group level and individual level. In addition, the whole brain functional network can be decomposed into several connectome gradients. The most dominant one, also known as the principal gradients, extends from the primary cortex to the transmodal cortex, indicating a gradually changed connectivity pattern from the primary to the high order regions. So how are these functional connectome features altered in psychiatric disorders? According to the survey of WHO, the mental disorders had the leading global burden among all kinds of diseases, and the prevalence is relatively higher for several major psychiatric disorders, such as the depression, the major depressive disorder, affecting over billions of people worldwide. However, the underlying pathological and physiological mechanisms still remains unclear. So in recent MRI studies focused on brain connectomics, researchers have discovered that most psychiatric disorders, from depression to schizophrenia, are closely associated with abnormal brain connectivity and faulty wiring within the brain networks. So this insight has led to the novel concept that psychiatric disorders can be classified as a kind of connectome disorders. This notion also holds significant value in unraveling the mechanisms and underlying psychiatric conditions and improving their clinical assessments. Accordingly, we have conducted many studies to investigate the alterations in functional brain networks in psychiatric disorders. So this is an early study aimed to examine the transdiagnostic alterations in functional connectome across depression, bipolar disorder, and schizophrenia. We collaborated with Professor Wang Fei and Professor Tang Yanqing in China Medical University. We collected resting state fMRI data from 512 subjects. There were over 100 patients for each kind of disorder. So we constructed the voxel-based high-resolution functional brain networks for each individual with over 50,000 nodes and over 10 million edges in each brain network. So this was mainly done based on our Pagani toolbox. We found a significantly lower local clustering coefficient and characteristic path length in all three disorders. So this suggests reduced local efficiency, but increased global efficiency of the patient's functional brain network. These results support the notion of the randomized configurations of functional connectome in psychiatric disorders. However, there are also differences among the three disorders, where the schizophrenia has the most severe alterations. We further found that the altered connectivity was distance dependent. The number of short-range connections were reduced and mainly connected to some primary cortex, such as the sensory motor and the visual cortex and the thalamus. In contrast, the number of long-range connections were increased and mainly connected to the prefrontal cortex, which is a brain region in charge of some high-order cognitive functions. These unbalanced alterations may contribute to the randomized network configuration. So we further examine the modular architecture in the 3 major psychiatric disorders. In accordance to the randomized configurations, the modularity was significantly reduced in all 3 psychiatric disorders, with several excessive connectors that are linking, that have more intermodal connections. So the disrupted modular structure is highly associated with the randomized connections. The hyperconnect connections were observed among almost all the functional systems in all 3 kinds of major psychiatric disorders. At the regional level, the over intermodal connections were mainly observed in several connector hubs, such as the dorsolateral prefrontal cortex and the medial prefrontal cortex. Interestingly, these regions are widely selected as target regions for neuromodulation techniques in clinical practice, such as TMS or TDCS treatment. Our findings provide an implication for their treatment efficiency from a connectome perspective. On one hand, these regions exhibited alterations in all 3 disorders. On the other hand, these regions were connected with various functional systems. Thus, so stimulus in these regions could affect a more wide range modulation in the brain networks. So, well, it should be noted that these analyses were conducted in data that were collected from only one single site. When we reviewing the finding across imaging studies, the results are not always consistent and sometimes are poorly replicated. An important reason is that the sample size of these studies is often small and was collected in a single site. So this could result in a large sampling bias in both demographic and clinical variables. In addition, the data processing strategies were also largely different across studies. About 6 years ago, we have written a review to highlight the importance of imaging big data for conducting functional connectome studies. Particularly, we emphasize that a large sample data size collected across projects or centers are greatly valuable to evaluate and improve the reproducibility of MRI studies. So following this idea, we initiated the disease imaging data archiving project in Beijing Normal University. We cooperated with several top hospitals and research institutes in China. For the MDD group, we have collected resting state fMRI data from over 2,000 participants. We established standardized archiving and management and data pre-processing procedures and quality control pipelines for all the data sets. So this data was shared among the participating institutes. This project is still ongoing and we are welcome for more collaborators. So with this large data set, we can improve statistical power to find subtle brain alterations related to disorders and examine the reproducibility of results across different centers, and identify some reliable structure and functional alterations in these brain diseases. Moreover, we can assess the effects of specific demographic or clinical variables in the disease and provide imaging biomarkers for diagnosis or treatment targets. For the methodology, these data could inspire innovative big data analysis methods and also optimize the scan procedures for future multi-center studies. With this data set, we first examine the reproducibility of functional alterations in depression regarding the pre-processing strategies, statistical methods, reproducibility across centers, and the effect of sample size. We examine the frequently used functional activity and the connectivity measures from very short distance to a long range distance. Generally, when we put all data together, we found that patients had hypoactivity in the primary sensory motor, visual, and the orbitofrontal cortex, and hyperactivity in some high order regions, such as the supramarginal gyrus, precuneus, and dorsolateral prefrontal cortex. While these findings were largely reproducible when we use different pre-processing strategies on global signal regression or filtering, or different statistical methods, this result suggests that the sample size may be more important than the selection of analyzed strategies. So for the raw data, we observed a very strong side effect on every functional metrics all over the brain. So this yellow region indicates between side difference on these metrics. So these results highlight the importance for data harmonization across centers. So methods like combat and some linear regression can effectively remove the side effect and increase the reproducibility among data from different centers. Actually, we are currently also developing some deep learning-based method to do the harmonization. Lastly, we estimated influence of sample size on identifying depression-related functional brain alterations. So we designed a sampling simulation analysis. That is, we randomly selected participants from each group of the dataset and examined the between-group differences. So the sample size ranged from 25 to 700, and this procedure was repeated for 10,000 times for each sample size. Then we can estimate the reproducibility as the percentage of times of identifying the same brain alterations. We found that at least 400 participants in each group were required to reach a reproducibility of 80% under a strict significance level. So this simulation provides an explanation for the sampling bias for previous small sample studies, and also indicates that there might be a large inter-subject difference among the patients. So therefore, we have much confidence that the large sample data set could help us to find some reliable connectome alterations at the group level. While depression is associated with the coexistence disturbance in both low-level sensory processing and high-order connectivity functions, and depression could occur as early as the adolescence. So because the principal gradient spans from the primary to transmodal regions, which are critically developed in adolescence and support both low-level sensory processing and high-order functions. So we focused on the disruption of the principal connectome gradients in depression. So for each participant, we decomposed their voxel-based functional network into connectome gradients. The principal gradients were organized along a gradual axis from the visual and sensory motor to the default mode network, which replicated recent findings in healthy adults. The inspection on histogram revealed that the extremes of the gradients in depression were contracted. And following the subsequent statistical analysis revealed that the depression patients showed a less explained variance, a narrowed range, and less spatial variation in principal gradients, suggesting a less differentiated connectivity pattern between these systems. And the local alterations were mainly involved in the primary systems that account for functions involving sensory processing and transmodal systems related to high-order combinations. And additionally, the altered principal gradients were correlated with a randomized network topology. We also conducted a connectome and transcriptome association analysis using the gene expression data from Allen Institute. The spatial pattern of gradient alterations were correlated with gene expression enriched in synaptic signaling and calcium ion binding. And the depression-related genes exhibited significantly higher weights in these multivariable models. We also found that patients with an onset in adolescence showed a more severe disturbance, suggesting an underdevelopment of the primary to transmodal gradient in adolescent patients. And the baseline gradient map can predict symptom improvement after an 8-week antidepressant treatment. So these findings provide a picture of how the functional brain network disrupted in depression at the group level. However, we also noticed that the effect size of the between group difference were almost less than 0.3, indicating there are a huge inter-subject differences among the patients, and potentially there might exist some biological subtypes. So next, we conducted an analysis to characterize the individual difference in depression patients. So we first estimate the functional connectivity strengths for each brain region for each participant. The FCS represent the sum of connectivity of a single brain region and has been proved to be the most reliable and reproducible measure of the functional brain network. Then, for each brain region, we charted a typical developmental trajectory for the functional connectivity strengths from 11 to 19.3 years old in health controls using the Gaussian process regression. This model can delineate the normative percentile charts for each brain region, which is similar to those height and weight chart for children. So for a given participant, we can estimate the deviation of the FCS of each brain region from the normative trajectory at any stage of the life. So we also conducted some validations like tenfold cross validation to ensure the reliability of our models. Then for each patient with depression, their FCS map were positioned on the normative model, and we can quantitatively estimate their deviations. This obtained a deviation map for each patient. On this map, we can know which brain region was the most abnormal region and also the extent of abnormality. Based on the individual deviation map, we can characterize individual differences among patients and further identify neurophysiological subtypes in depression. So at the population level, we found that patients had an overall greater deviation than the controls, mainly as a result of increased connectivity strength in the default mode region. However, when we analyze the data at the individual level, we show that only about 70% of the patients showed an extreme deviation in at least one region. And for any single brain region, no more than 4% of the patients showed extreme deviation from the normative range. So this suggests that the specific brain region with significant FCS change vary considerably from patient to patient. So using clustering method, we further identified two subtypes in patients. The optimal number of categories were determined by a series of evaluation indices. About 37% patients were classified as subtype 1 and 63% were subtype 2. Importantly, although with fluctuations, this ratio was largely reproducible in each site and showed an overlap rate above 90% in leave one site out validation, suggesting this subtyping would generally existed in different sites, but not driven by biased sampling among the centers. The subtype 1 had severe deviations with positive deviation in the default mode, limbic, and the subcortical areas, and negative deviations in the sensory motor areas, while the subtype 2 had a moderate abnormalities with an inverted pattern of deviations. So after subtyping, we observed a higher consistency of extremely deviation regions among the patients of subtype 1. So the following clinical validation showed that patients in severe deviation subtype are generally older and have higher proportion of medication and have more severe symptoms in the suicide item, while the moderate deviation subtype had severe symptom in the work and activity and the depressed mood items. This implies that patients with more severe brain alterations may associate with relapse and extremist thinking. More interestingly, using the baseline deviation map, we can significantly predict symptom changes after 6 months of antidepressant treatment. However, this model only works for the severe deviation subtype, but not for the moderate deviation subtypes. So this might be because of the large imaging variation in subtype 1 and suggests that different clinical assessment strategies should be considered for different patient subtype. We also conducted some numbers of validation analysis. Fortunately, all the findings remain stable. So in summary, we established a number of toolboxes for MRI data processing, data harmonization, brain network analysis, and visualization. We reviewed transdiagnostic randomized configuration and disrupted modular structure in the functional brain networks across 3 major psychiatric disorders. So with the multi-center collaborations, we found reproducible functional brain alterations in both primary and high-order brain regions, and downgraded and contracted principal connectome gradient in depression. There was a great heterogeneity in functional connectome deviations in depression, inspiring 2 reproducible neurobiological subtypes, which differed in imaging and the clinical features and treatment outcome prediction. At last, I would like all the collaborators and my colleagues and the students at Beijing Normal University, and also I would like the support of our fundings. That's all for the talk. Thank you for listening.